Hello. Well, welcome. How's everything going today? Sushmanth, do you want to give an update?
Hi Bradley, how are
Hi,
you?
how are you?
Uh, Bradley, I was working on the GSoC submission itself. My
Okay.
deadline is on October 23 next week. So I was preparing the documentation, etc. I have been pushing some code actually into the DevoWorm repo. I think you have saw it.
Yeah.
And I haven't pushed any code of DevoNet. I'm working on it actually. Not maybe, I will push the code. I want to keep that repo private because there are some things which I want to add and I want to release completely after working on this because we are writing a paper. Before writing a paper, should I keep it private or I don't know. But we can keep the cell completely public for GSoC submission itself, and I will push the complete code and with the perfect documentation. I was adding tutorials, etc. I'm also writing a separate blog for fine tuning SAM, how to fine tune SAM on microscopy images, not only for C. elegans, etc., other organisms also. So people can use that documentation, and I am creating a separate documentation for GSoC submission, and these repos will be updated over there. And DevoNet also, I will push the code broadly, but I just want to keep it private until the perfect tutorials, comparisons between different models will be hosted. Is that okay?
Yeah, that sounds good. That sounds good.
Now you have some pretty different picture on the whole project. By next week, everything will be ready with the perfect documentation. I'm right now working on it only. So I will show you in the next week Monday meeting, October 23rd. Whole project, what I have done in the GSoC period. Maybe I will ask Mahit to join that meeting and I will show him to him.
All right, that's good. Yeah, you should be here to see what it's, we can make some comments on it. But yeah, I think that's great work that you've been doing. I saw the submissions in the, I saw the pull requests and all that, so that's good.
Okay. I
Yeah.
have been pushing code. It is not completely added badly. There are some notebooks if you want to see around what I have worked on. But there is a folder called notebooks in it. There are like how I handle the data and the loss functions graphs, etc. are there. And the inference file also is there. How to use our model for different type of microscopy images is also there. So people can use it. Right now it is in draft position only. By next week it will be completed. And next week you and Mayuk can discuss it further and you can take a decision. But next week I'll ask Mike to join the meeting. He will join. Actually, he was having a graduation. He attended to the college. Maybe next week for the meeting he will attend this meeting, Monday meeting also. He will evaluate completely. What he want to add some suggestions also. Right now tests are failing in DevoWorm library. When I met him 2 days back, he mentioned me like you need to work on that issue. Create an issue and work on that also. He told after this GSoC submission, I will work on the DevoWorm failing test case.
Okay, yeah, that's good. I know things can fail. Yeah.
Yeah, I will create the documentation perfectly. Right now, I was doing that only, creating a perfect documentation. Whole code is completed. I'm just creating the documentation and etc. things to follow. So I will leave the meeting and I will give my complete update on the October 23rd meeting because we have only 1 week. I need to do a lot of documentation for that. Is that okay?
Yeah, that's good. Yeah, and congratulations to Mayuk for graduating.
Yeah, thank you.
Yeah.
I will let him know.
Okay, yeah.
Tomorrow we have a meeting. I'll let him know.
All right.
Thanks, Bradley. I will
Right.
leave the meeting. I have these things to follow up. I will attend next week meeting and my submission deadline is on October 23rd.
Yes.
I'll submit it and parallelly you can evaluate and by October 31st, I think so. It was showing as October 31st to submit your evaluation, mentor evaluation.
Yeah. A week after the submission. Yeah.
Okay. Those are my updates, Bradley. I will let you know in the next week meeting. I will share some communication Slack over this week because there are a lot of documentation I want to show you before submitting to the final submission. I will chat you over the Slack. Is that okay?
That sounds good. Yeah.
Okay.
All right.
Thanks for me. I will leave over the meeting. I have this work to do. Okay. Is that okay?
Yeah, it's fine. Talk to you later.
Thanks.
Bye.
Thanks. Bye.
Okay, that was Sushmanth's update, and looks like he's coming along on his project, and he's going to be ready to submit by next week. So next week he'll be doing a formal presentation on his project, and hopefully Mike will be here. He'll be able to talk about it more then. So, Susan, how are you? Are you available to talk? And
how am I? Well, I'm fighting with models. I've got, yeah, I'm just, I need to get my graphs in order and write it up. So my models are not behaving themselves well because they're tensegrities, but I'll just, whatever comes out of the computer, I'm just going to draw a graph of it and hand it in right about now. Doesn't matter whether it's crooked or not. Yeah.
Are you going to draw it by hand, or are you going to draw it in the...
Oh, no, I'll put it into MATLAB, and it can draw me a nice graph with a title. And
oh, right, yeah.
Yeah, and then I can talk about it, and maybe I can get 10 pages out of it, and since I have 18, then maybe I can squeeze 30 pages and hand that in to them. This is revenge of the nerds. Hand out 30 pages of a write-up. What? You thought I wasn't doing anything? Well, here you go.
Yeah, well, it's... Yeah, fighting the good fight with models, you know.
Yeah, well, models... anyway, it's interesting how they change when you change their attachments. So I think that's what I'm going to focus on. See, it changed when I did this, changed when I did that.
Right.
Because in a cell, it's important, the base membrane that they're attached to is very important to their development, because if you have a soft substrate, then you get nerves. If you have a really hard substrate, then you get bone. I actually have I might have shown you it before, but I have a nice paper and it shows that.
Okay.
So I can quote it and I can say, see, this happens with the model. You know, so that's a good outcome, that even if the graph has got weird blips.
Right.
Anyway, so that's me. I just need to get this done this week.
Well, that's great. Yeah, it sounds like you're getting somewhere.
Yeah, I'm getting somewhere. It's just not quite what I want it to do, but yeah, it's experimental. So why wouldn't it be misbehaving itself? No, it doesn't work quite as smoothly as I would like to say.
Yeah.
That's all.
Well, that's good. Yeah. So good luck with that. And you asked me about this. You said, is there any data on epithelial cells in terms of their size or shape? And I looked a little bit and I didn't see anything, but we'll talk about that a little bit later, what people are doing with maybe getting somewhere towards that.
I have permission from Professor Zhang. He's one of the advisors. He just said, look it up on Wikipedia, average cell size, just make some average cell sizes of different shapes and then just call it a day. So I'm going to do a flat one and a tall one. And they're going to be different volumes, but I'm just going to do it that way and then say it would be better to compare ones that are of the same volume, and it would be nice to have references, but there aren't. So the only thing I can find is the lens of the eye, the cells in the lens, size, exact size. I don't know. It's being hard to look up. And there's no tables, like no repository for cell sizes.
Yeah.
Like it depends on the species and where in the body a cell is as to its size. But okay, so, but there's no table, no repository.
Yeah, I know it's like a common problem. Like I've seen in the literature, they're just kind of at the level of characterizing different cell types in terms of their numbers, which is an estimate, but like getting the sizes for every tissue and every cell type. I mean, there's a lot of diversity there that you can't really just say.
Yeah, well, but even if I had one thing that said mouse intestine islets or something, right. I don't know. But then I could say, okay, well, this is a cell and it belongs here and it's this size. And so it behaves like this. Like, anyway.
Right, yeah.
There is no such thing.
Yeah, it's not, they don't have that kind of, they don't sort the cells and count them like that usually. Usually if they're doing that, they're trying to get molecular data, so.
Yeah, they can probably tell me what proteins it has in it, but not the size of the actual cell, which they've imaged on the microscope, and they know what it is, but they just somehow it's not
in the literature.
Put in the literature, and then you look for the images, and they forgot the scale bar. The scale bar isn't there. So,
yeah.
Okay, I give up. Anyway, yeah, that's just me. Anyway, enough of that. It's just, I was given permission to just use a generic cell size and carry on.
Okay. Anyways, yeah. So yeah, other news, we had our paper that we were working on earlier, and that's the one we submitted to Biosystems. And of course, that got bounced back with some comments, and we have to resubmit it to Biosystems. So Dick was in conversation with some people at the journal, and we got another chance to submit it. And I'm going to work on that, but not like this week. I've got a lot of things going on right now. I'll try to get to it maybe by maybe 2 more weeks or so. I have a couple things deadlines coming up I need to make, but we'll start working on that soon and revising it. So there are some things that I'll probably work on with Lucas, and we'll get there. It's a pretty good paper though, I think. It's just, it's not quite palatable to the larger community. So we'll figure it out. Yeah. So Dick or Jesse, anything you want to talk about before we go on? Okay. Well, so I mentioned before that. I mentioned before that I did a little bit of looking in terms of the epithelial cells, and no, they don't have those specific measurements. Okay, thank you, Jesse. Well, they don't have those specific measurements of size and width and everything. They do have some characterization of the phenotypes of cells. So like I said, a lot of the cells are, there's a lot of diversity in, like, say, the human body in terms of epithelial cells. In other species, epithelial cells, there are a lot of different subtypes and things like that. But we do have some sort of move towards characterization. I mean, there's no one, place where you can go and find all this information broken down by cell type. I think a couple weeks ago, we saw a paper where they had done a count of different cell types, and those are all estimates, really. But they did a little bit more rigorous job than previous studies, largely because they did a survey of the literature, and that was kind of how we did that. So let me share my screen. And I'll talk about what I found in terms of the epithelial cell characterization. So this is a picture of some of the different types of epithelial cell and different types of kind of tissue that they're embedded in. So this is, I think in humans, this isn't going to show up well. It's kind of hard to see. Let me zoom in a little bit. It doesn't work like that. Okay, we're just going to have to bear with this. So there's a, this is a table with some images on the right, in the right-hand column. This is epithelium and different types of epithelial tissue and cell. And then this is the identifying features, the function, the location, and then this is the site. You can see that these cells right off the bat are very different in size and shape. So some of these are square. So the ones in the stratified cuboidal type cells are in the ducts of the mammary glands, sweat glands, salivary glands, and pancreas. And you can see the basement membrane is here. And then these are the layers of cuboidal cells. So we have two layers in this image, which they're basically around the same size. So, you know, they're stacked on top of one another. They're involved in secretion and absorption. And they usually consist of 2 to 3 layers when you see them in a histology or something like that. That's just one type of arrangement for the cells, one type of cell, stratified cuboidal. This one here is stratified columnar. This is where we have several layers here. The top layer being a lot longer or lengthwise or height-wise than lengthwise. The length being across and the height being up and down. So you see that the top layer here is a little taller. The bottom layer is a little taller than the middle layer. So these are distinct types of cells. I really cannot zoom in on this without like ruining the background. So I can't really make them out too well, but these are just different types of cells. And they exist in these layers. So these are multiple layers of cells. Basal layer is usually cuboidal. So that's the bottom layer here next to the basement membrane. The top layer usually is columnar cells. So this is a different cell type. And then this middle layer is a different type of cell. So they're even in the same sort of structure, you have a heterogeneity of different types of epithelium cells. So this is transitional here, multiple layers of cells. Basal layers of cells are columnar or cuboidal, so it's a mix again. Intermediate layers are pear-shaped. So it's really hard to see this, but you have like these you know, the top layer, the bottom layer, and then the middle layer where the cells are more pear-shaped. Top layer has umbrella-shaped, dome-shaped cells. So you get this kind of effect where they come out and bulge out at the top. And then this is usually, you find this in the urinary bladder. So these epithelial cells are found in the bladder. And they're very different from, say, what you find in the glands or in like the pancreas. This is the stratified squamous non-keratinized type epithelial cell. We have multiple layers of cells. The basal layer, which is down here, has columnar or cuboidal cells. And the intermediate layers are polygonal. So you have these polygons. And we talked about polygon-shaped cells. In Archaea, you have polygon-shaped cells in some structures in like the human body. These are polygonal cells. They're just kind of like different shapes. And, you know, they're, you can see here, it's really hard to see the actual shapes, but there are a variety of shapes in here. And then the top layer here, up here, are flattened or squamous cells. So these are flattened cells that have like a short height and a very long width. So this is, you know, but you have this kind of structure. These are usually found in the esophagus, tongue, and vagina. Again, it's a functional difference in terms of what these cells are doing, the structure that they make. but you have these multiple types of cells. And again, this is this is a protective layer. So you have, the imperative here is to maximize structural integrity. Finally, at the top, we have stratified squamous keratinized cells. And these are for, again, protective. This is in skin epidermis. So this is the surface of your skin and right below. So, obviously that's a protective, there's a protective component to skin. And again, it's multiple sort of types of cells, multiple shapes represented. The identifying features are multiple layers of cells from basement membrane to top. So it's from the bottom to the top. We have the following types of cells. Number one, the basal layer at the bottom, which is down here, has columnar or cuboidal cells, again, like the stratified squamous non-keratinized. Number two, you have many layers of polygonal cells in the middle. And maybe you can see them a little better here, but you have these different shapes in the middle here. They're not quite diamonds, but they're kind of like these, they're not quite square either. They're kind of blobby, but they have a polygonal aspect to them. So, they have some sort of shape.
Are those the ones that they're kind of squished together like soap bubbles?
Yeah, I think
like
so.
That, right?
Yeah.
So it goes from your honeycomb shape to kind of over the layers and back into the soap bubble shape type cells.
Yeah.
Yeah. It would take a lot to model that.
Yeah, I know.
In hours.
You got different cell shapes, different cell types, probably different types of, like you at the basement membrane, I'm sure has a different set of physical properties than towards the top. In terms of,
Yeah, different attachments.
Right.
Yeah, different trijunctions.
Yeah.
At the corners.
Yeah.
All kinds of interesting things. Yeah, trijunctions are interesting. They've managed to find, their composition. And they're fairly recent, like they're 2023, 22 type references that I have. So it's a new area. What is a trijunction? Where three cells meet.
Yeah.
Yeah.
Yeah, we talked about that outside the meeting, but yeah.
Yeah. Okay.
So yeah, so we have the flattened cell, or we have polygonal cells in the middle, and then at the top we have flattened cells filled with kertohalline granules. So that's these darker cells at the top. So there's something, it's more of a protective layer here. Whole thing is protective, but especially at the very top. And then the topmost layer-shaved dead scaly cells with the out nucleus. These are filled with keratin. So at the very top, you have these cell types that are just kind of like keratin-filled. They're dead. They flake off. That's why your skin flakes off. And it's like kind of, I don't know if things are moving upward in the columns towards the surface, like if there's some sort of trophic cycling going on within the skin where the cells move up and out. But there's this layer is constantly, cells are dying and being replaced. And so this is all for protective function. So that just shows you the diversity of cell types and structures that you can have with epithelial cells. Polyhedral, not polygonal. Probably polyhedral. It said polygonal in the figure, but probably polyhedral. Let's see. The next thing is this figure here. So these are types of epithelium. So these are the different types. You have the simple squamous, which is the single layered. And you have these polyhedral cells in here. You have the simple cuboidal, which are these cube-like cells. And again, these are all very much, you can probably get a good average shape for something like this and not be, too far off. Simple columnar, which is kind of like the cuboidal, except it has a greater depth. There's stratified squamous, which is where you have like kind of the simple columnar layered on the basement membrane, these polyhedral cells in the middle and the top, and then that's it's a kind of a compound structure. And there's stratified cuboidal, which is like the simple cuboidal, which is stacked. So you have multiple stacks of these cuboidal cells, multiple layers. You have the pseudostratified columnar, which is where it's interesting because the simple columnar is very much kind of like, regular, sort of a regular size across cells. So you could take this and say the average size pretty much characterizes each cell. In this case, it looks like there's some sort of pseudostratification going on where it's almost like you're taking a cuboidal cell and you're stretching it out and you're getting like a wider width up at the top versus at the basement membrane. So you get these differences in shape, or they get these differences in shape due to sort of the way that they're layered in here. They don't form two distinct layers, but they have like, some of them are sort of in here, wedged in between these two cells. These two cells have stretched out above this cell and form kind of a second layer, but it's not formally a layer, which is, kind of an interesting geometry because, again, that's going to be something where maybe there's a bimodal distribution of average size. So, and then there's the transitional, which is where we have the basement membrane, and then the top. In the top, of course, the cells are the sort of, they're not really squamous, they're not cuboidal. They're kind of maybe cuboidal, I guess, but they're very large compared to the ones below it, which are smaller. And then you have these columnar cells at the bottom, which are long and thin. And, again, how would you get the average cell size here? There may be different modes of that distribution where you can get an average for each cell type. So yeah, it's pretty hard to model that because there's just so much variation there. This is actually kind of the picture that we had before. Well, this is a different picture actually. And this one talks about, let's see, okay. So this is where they kind of show these types of epithelium. Then they have these types of, so these are different types that I showed from the other one in different images. So these are microscopy images. There's simple squamous. You see this in the alveoli of the lung. These are, the function is that they form a thin layered surface, and they're good for diffusing gases. The cells are large and flat. The next type is simple cuboidal, which is where it keeps shape, but it also needs to absorb liquids and things like that. This is something you find in the kidney tubules. So you see these cuboidal cells, but they form these rings. And so that's, they form these larger structures. They have functional significance. The simple columnar, this is large surface that leads to absorption. So they're arranged in this sort of large surface area. This is where you have these columnar cells that are arranged in a sort of a oblong shape. And this is found mainly in the intestines. And these may have microvilli, so they have additional functional aspects to them. There's the pseudostratified ciliated columnar epithelium here. This is where you have goblet cells that secrete mucus and move dust. So you find these in the linings of the respiratory tract. And they look like, this is where we have this sort of pseudostratified type of columnar cell where it looks like multiple layers, but it's really not. The cells kind of are these globs that sort of sit together. Sometimes there are some that are displaced and sort of at the top or the bottom. Others just kind of have a variable shape across the column. There's transitional epithelium. You find that in the bladder. So that means that you have different types of cells arranged into this structure. This allows it to stretch easily. You have the stratified squamous epithelium as the final one. where you find this in both the skin in terms of keratin and the tongue in terms of the species, whether it's keratinized or not. So in cats, the tongue is keratinized. In the humans, it does not have keratin. But this is a protective layer where the surface is likely to suffer friction. So especially in a cat's tongue, they're always licking things. You have friction there all the time. You want to have a protective layer. And so in this case, we want this stratified squamous type cell.
Do you have that reference? These are nice pictures.
Yeah. Yeah, I have them. Oh, go ahead.
Yeah, where did you get that from?
Yeah, I have to find the reference. I think it's in here somewhere, but I'll send you this stuff after the meeting.
Okay, well, thank you. Those are nice images.
Yeah.
Again, they don't have a scale bar.
Well, yeah. They're kind of like examples. They don't like put the scale bar on them. But, yeah. I don't know what the scale would be, though. I guess it would be like if you had, well, it'd be kind of hard to reconstruct that because...
Depends on the species and the place in the body.
Yeah. Then you'd have to...
That's where it is, but, is it a mouse?
You have to measure the... Oh.
Well, anyway, I still don't know what the size of human intestine columnar cells are. Like, I don't know what that is.
Right.
So, even though I looked, I spent a whole day on it. But anyway.
Yeah.
That's enough time to spend on it.
Yeah.
It's good enough.
Yeah.
Like I said, he says, just go with what Wikipedia says and take an average and just do that.
Yeah.
For initial purposes. I could refine it later.
Right. I think probably you get a really to get an accurate measure would be like, you could still do an average, but you'd have to have like the area of the structure and then what, how many cells are there, which is really hard to do. Or if, they're, just taking that little area. I mean, if you had a scale bar, it'd be nice, but...
the problem is they show the surface quite often, and they're not showing the depth. Like, I need to see the surface and the depth of the cell.
Yeah.
Both. Kind of at the same time. Like, taken close together, the side and the top of the cell in order to find the cell area, cell volume,
right.
Or even just height and width would be helpful. Anyway, it's elusive, partly, I think, because the microscopy is usually looking down on it from the top and not measuring the depth.
Yeah.
Which is an interesting thing because we're working with optical coherence elastography, which does see depth. So maybe one could put together a whole catalog of, these cells are about this width and this depth. Like, I don't know.
Yeah.
Even samples that are dead you could do this with. Like
right.
It could be a whole area of study.
Yeah. Size characterization or something. I don't know.
Yeah. Anyway.
Yeah. So there are two papers here. This one is actually about the imaging. So this is imaging the cell morphological response to 3D topography and curvature in engineered intestinal tissues. So what they're doing here is they're trying to develop a method for looking at the tissues and looking at the depth and some of the curvatures that we see. So conventional cell culture methodologies have relied on flat two-dimensional cell monolayers. Three-dimensional engineered tissues are becoming increasingly popular. So this is why we need this. Engineered tissues can mimic the complex architecture of native tissues. Here we examine the cell morphological response to these villus-like microstructures at single cell resolution using a novel embedding method that allows for the histological processing of these delicate hydrogel structures. So they're in like this in vitro hydrogel, and they're going to image it there. But still, they're trying to get this depth and curvature aspect. We demonstrated that by using polymerisable polyethylene glycol as an embedding medium. So they put the cells in a medium. The villus-like microstructures were successfully preserved after sectioning with vibrotome or cryotome. So they sliced it up. Moreover, high-resolution imaging of these sections revealed that the cell morphology, nuclei orientation, and the expression of epithelial polarization markers were spatially encoded along the vertical axis of the villus-like microstructures, and that the cell morphological response, which is this response to, I guess it's just being in its place, was dramatically affected by substrate curvature. So this is like, let me see if they have any images in this paper where they kind of talk about what they're using here. Actually, they do, I think they mentioned, oh no, they don't. But this is their setup here. You have the substrate here, the UV source, the photomask. They have this. This is just so they're fabricating this these scaffolds for the microstructure. Yeah, they're villus-like microstructured hydrogel scaffolds. So this is what this looks like here. Then this is bright field microscope image of the cross section, and then this is the confocal maximum intensity projection showing nuclei staining and F-actin staining in a certain type of cell here, grown on top of the villus-like scaffold. So this scaffold is here. They're growing the cells on top of it, and they're able to get they're using a confocal image to sort of show the cells, and they're expressing their markers, and then they're going to grow them on top of the scaffold.
Okay, they're trying to get at 3D there, which is nice, but still not like the tissue. It's their individual cells, so.
Yeah.
But anyway, yeah, this is a nice attempt at doing that, so that's fine. I still think my infrared light might do a better job.
Yeah.
Yeah, because you can get it as fine as about 2 micrometers, so you could get that sort of imaging, I think.
Yeah.
Anyway, next comment.
Yeah, so this is, let's see. Yeah, they just show kind of some of their proof of concept here. They don't really show much. Yeah, they just show kind of like some of the layers and what they're the kind of images they're getting. Still, I don't think it's at that single cell resolution that I mean, there is some single cell resolution here, but not at the kind of resolution we would like. Just it's showing. Well, in this case, they're just showing the different markers. So beta catenin, they're showing a nucleus stain, and villain. So it's like there's these things that are being expressed in the cells, but not like the actual enough to, I think, get the information for shape that we would like. Okay, so that's that paper. Yeah, I'll send these papers to you. And then this paper here is dynamic changes in epithelial cell morphology control thymic organ size during atrophy and regeneration. So this is where they're looking at a specific problem. They're looking at T lymphocytes and how they need to be produced through life. Yet the thymus, where T lymphocytes are made, exhibits accelerated atrophy with age. Even in advanced atrophy, however, the thymus remains plastic and can be regenerated with appropriate stimuli. So this atrophy is thought to reflect senescent cell death, while regeneration requires proliferation of stem or progenitor cells, although evidence is scarce. So these conditional reporters to show that accelerated thymic atrophy reflects contraction of complex cell projections unique to cortical epithelial cells, while regeneration requires their growth. And then atrophy and regeneration are independent of changes in epithelial cell number, suggesting that the size of the thymus is regulated primarily by rate-limiting morphological changes rather than by cell death or proliferation. So to get there, they do some characterization of the cells. They look at changes in cell morphology. They have this, C is the network map of different GO categories. So you're looking at genes here, gene network, and you're looking at these genes for these different mechanisms here. This is their microscopy. I don't think it's really relevant to getting size and shape information. They're able to characterize the cell, the cortex of epithelium here. They're able to get some information about the cells. They've stained them, but I don't see any. They have so they have total surface area, ferret diameter, and occupied cell volume. So again, this shows at different stages here. I guess this is in development and regeneration. Let me see if I can find the legend for this. Here it is. So these figures here, K, L, and M, are these here. So this is K through M, measurement of relative physical parameters in CTEC, such as total surface area, ferret dynamic, or cell volume. And brackets indicate statistical significance. Biologically independent tissues, each for 5 weeks, 12 months in regenerated groups. And so they talk more about that in the data file, but basically they're getting total surface area, microns squared per 10 to the negative 3. This is ferret diameters in microns, and then this is occupied cell volume in microns to the third. This is volume times 10 to the negative 3. So you get the surface area squared, cell volume cubed, and this is what it looks like here, this distribution. So you can see as you go from 5 weeks to 12 weeks, it drops off, and then in regeneration, it resembles more the 5-week condition. Beyond this, I don't really know much about the data, so that's something that maybe we can look up later and take a look. But in this case, they're at least able to show a little bit about characterize the sizes a little bit. But that's not an ideal graph. But that's basically the sort of scale we're working on.
They have scale bars, which is nice.
Oh, in these images, in these images here.
Yeah, in the images they have scale bars. So yeah.
So this is actually so they're looking at atrophy here, and they actually have a couple of nice images in this figure 3. So this is actually E, maybe from D to E. This looks like some sort of analysis here. Orthogonal projections of the 3D distribution of MTEC shapes from young red or old blue mice. So these are young mice in red, old in blue. You have this 5-week and 12-week, or 12-month, I'm sorry. I think before I said week and it's really month. But so these are young and old, basically. So we have these different shapes. So these graphs here in D, we have compactness in this axis for this image. We have compactness on the x-axis, volume ellipsoid ratio on the y-axis. So we have this volume versus ellipsoid, which is where they characterize the sort of the elliptical nature of the cell. This is the ratio between the two. And then this is compactness, which is another measure. They have these different components of this. So the idea is they overlap these, and they want to see the degree to which these two sets overlap. So for the old mice and the young mice are further in this direction than the older mice. The older mice are a little bit back in this direction. So there's some difference in the volume ellipsoid ratio. It's a little bit higher in the younger mice. But overall, these overlap pretty well. Down here, this graph is ferret diameter in microns versus compactness. So the compactness is on the y-axis now, and we're looking at the diameter versus compactness. So that's, this is, they say at least 1,000 objects are shown for each age. So that's pretty good, pretty decent amount of data for that. And so this, in this figure here, E, F, and G, these range from larger and extensively branched to smaller and non-branch morphology. So what they've done here is they've captured some volumes, and these are just sample volumes. So they're not like a huge database, but they have a scale bar, as Susan said, and so they're on the order of maybe 15 microns. I'm not really sure what if you were to like measure it out, but I don't know if these may be in the data, in the supplemental data too, which would be nice because that would actually give you at least something. Even if it's this kind of a characterization, it'd be interesting to look at. And so then that's what they're kind of working on this specific problem in this specific tissue, but that's what we have. That's like one of the better examples of what we have. So then they just do a genetic analysis here, which is like talking about cell cycle genes, and if you're talking about regeneration, or you're talking about apoptosis, then you need to think about cell cycle genes, and that's what they do there. And so that's that paper. And so that's what I have on that. I picked some things that I found. It's not by any means is it a comprehensive review, but I just wanted to see what was out there in the literature. So I'll send you, Susan, all those references, and I'll try to get references for the images. I'm not quite sure where they came from now, but I'll get them. Yeah.
Well, thank you. That's okay. Don't go to too much trouble. I just need to write this up using Wikipedia. Says that cells are between 110 microns or 10 to 100 microns, and I'm doing some that are like 10 micrometers wide and 100 micrometers tall for a columnar cell, and I think they'll figure that's all right.
Yeah, probably.
So yeah, it's just preliminary. It says preliminary data. He put preliminary in the section, so this is preliminary.
They really want you to do this further, study it further.
Yeah, they probably do. Yeah, I'm supposed to be studying this, but anyways,
yeah,
just need to write it up.
That's great. Yeah. Okay. This is another thing that I found this week, and this is something I know Morgan is interested again in, these organoid models. We've talked about organoid models in the past, and so I'm going to share my screen again. And so there's this paper that came out, I think it was recently, 2023. It was in Seminars in Cell and Developmental Biology, and this is about mapping and exploring the organoid state space using synthetic biology. So there's this idea of the state space that we can produce to talk about some of these things. So we have all this variation that exists in different cell types and different tissues, and how do we characterize that variation? How do we know, especially in an organoid, which doesn't necessarily map to the differentiation patterns of real organisms. We just build these organoids, we try to differentiate them using chemical signals, and then we get these different cell types that maybe resemble some organ. And so, but how do we know what we're going to get when we create these organoids? What kinds of different cell types, what kinds of states can we expect to encounter when we do this? And so this is an emerging area, because organoid biology is sort of, people are just trying things and throwing them at the wall and seeing what works. So this is an attempt to sort of parameterize this process, which is, daunting, I guess, but they're going to try it here. So the abstract reads, the functional relevance of an organoid is dependent on the differentiation morphology cell arrangement, biophysical properties. So these are 4 things, at least, that we need to worry about when we think about the functional relevance of organoids. So we have these cells that we're differentiating into different cell types. Maybe we start to get layers of different cell types interacting with one another, forming a sort of a tissue or an organ of interest, or at least that's what it looks like. And then, we know that, like, there are these different things going on, biophysics, arrangement of cells, the morphology of the organoid, and differentiation of the cells themselves. So these are things that collectively define the state of an organoid. So for an organoid culture, so we're thinking about the cell culture as a whole, an individual organoid, or the cells that compose it. So you have the organoid, you have the individual cells. These are, the state of any one cell doesn't necessarily reflect the entire state of the organoid. So these state variables can be characterized most easily by transcriptomics and by high content image analysis. So you have microscopy, different types of microscopy, and transcriptomics is how they usually characterize organoids. So they sample like a single cell, they do some single cell transcriptomics, where they'll do like microscopy on the structure to see what it looks like. And maybe things are being expressed and you have reporter genes in there that give you some indication of the spatial distribution of what cells are doing what at what time. Their states can be compared to their in vivo counterparts. Current evidence suggests that organoids explore a wider state space than organs in vivo. So this is sort of the lesson we knew already, just based on the fact that organoids are these approximations of organs, and we can shove them around with different chemical media. So, if you change the chemical media, you can get it to differentiate more or less. So we kind of knew that they had this wider possible state space than an organ. So if you had, like a brain organoid, that brain organoid would explore more states as a tissue, its constituent cells and the interactions between them, than an actual brain in development. And that's what they're talking about here. This is due to the lack of niche signaling and the variability of boundary conditions. So this is where, again, lack of niche signaling means that you don't have these, niches within the tissue type that usually exists in a tissue and context. And then the variability of boundary conditions just means that it doesn't have the typical conditions of being in a body where in a brain, say for example, you get these connections to other target tissues that determine whether the, certain cells are, survive, or whether they apoptose or whatever. So there are these boundary conditions of being connected to the rest of the body that are important. Using data-driven state inference and in silico modeling, meaning that they just basically took some data, they tried to recreate states, and then they did some modeling in a computer. Phase diagrams can be constructed to systematically sort organoids along biochemical or biophysical axes. So their argument is that most of the variation in the state space is either biochemical, which means it's due to some biochemical interactions between the cells, or the state of the biochemistry within the cell, or the biophysics, which is the physics of the organoid, the physics of the cells manipulating or interacting, and the cells migrating and so forth. These phase diagrams also allow us to identify control strategies to modulate organoid state. So they actually can kind of identify things that you can do to improve control over the organoid and how it ends up, in what state it ends up. To do so, the biochemical and biophysical environment, as well as the cells that seed organoids, can be manipulated. So the cells that seed organoids are usually stem cells that come from some source, and you put them in the, you seed the culture, and then you get them to divide and differentiate, and they form these. So the idea would be to manipulate both the biochemical and biophysical environment. So that may mean that you have some sort of scaffold you're growing the organoid on, or you have the chemical medium optimized. And so that's what they're talking about. So, organoids are three-dimensional structures. People have been able to develop things like optic cups, intestinal organoids, mammary glands, and other things that, have some shape, defined shape and function. And so, these are things that we're getting more sophisticated with the design of these systems. So we need to have an idea of the state space. And so this is interesting. They talk about epithelium over here. So they talk about the tissue architecture. So these different states, they go back to Waddington, and they draw an analogy with the Waddington landscape, which we've talked about before, which is this landscape that has, these valleys and these canals, what he calls canalization, where, you start at the top and you follow these channels or canals down the landscape, and you end up in a final state that's sort of distinct from other states. Those channels branch off at certain points, so it's a branching process, and you go from undifferentiated cell at the top to differentiated cells at the bottom, and you can also talk about tissues in that way. So they kind of map the organoid state space to this Waddington landscape. And so there are these different trajectories in the landscape that define different outcomes. And so they talk about in the organoid state space, a state is a more complex object since it includes morphology, cell type composition, spatial distributions and patterns of the cells, and tissue architecture. So this is where they talk about in the epithelium, where you have, different types of epithelial structures, as we saw earlier in the meeting. When you talk about an epithelium, there are many states within that one type of tissue. And so, this is something that needs to be captured by the state space. And you can use a Waddington landscape to kind of describe those changes in this transition from undifferentiated cell to a differentiated state. So this is where, this is kind of where it gets, where they're trying to go with this. And then within this landscape, which is the Waddington landscape, I guess, normal organ development in vivo will correspond to a specific trajectory. So this is, in normal organs, they follow a trajectory on the Waddington landscape. But in vitro, in organoids, growth will respond to a set of nearby trajectories. So in other words, if you have an in vivo tissue type that's been sort of under normal conditions, it will have a single trajectory. It won't be as unstable as an organoid, where an organoid, if it's trying to form the same tissue, may form many different sort of variations on that tissue, and it corresponds to different nearby trajectories on the Waddington landscape. So the Waddington landscape is more fractitious. It's because you don't have those boundary conditions, and you don't have the context of the normal body. You have, you're kind of playing around with the medium. You have different biochemical noise, basically, and then you also have biophysical noise. And this is why you end up with this, larger variation than you would get in a biological context or in an organismal context. So they talk about morphospace, which is sort of an analogy to this Waddington landscape, which is basically where you take any morphology and you put it into a n-dimensional space, and you want to be able to define how different two objects in an organoid culture are. So morphospace is something that I think we've talked about in previous meetings. It's an allied concept to the Waddington landscape, and it's basically where you have a cell and it has different attributes, and you summarize those attributes in like maybe a three-dimensional cube, and each cell or each tissue type will have a distance, and that distance is determined by the factors that you're putting in, and there's the relative value. So there would be a difference in state based on this distance. So we have a morphospace, which is distinct from the Waddington landscape. It defines how different two objects in an organoid culture are, and then you can from there go on to describe the states in that state space. So this is where, it's kind of tricky because, you still go back to that issue of the state space is defining states. What are these states? And the answer is that it's these different trajectories on the Waddington landscape, and then the distance along this morphospace. So if there's a distance between two things and different trajectories in the Waddington landscape, then it's a different state. But this is something they're kind of working out. So this shows kind of what they're talking about a little bit more. This figure actually summarizes a lot of this. So this is where they're mapping the organoid state space using transcriptomics, high content imaging, and mathematical modeling. And in A, you have some organoids, and they're in this three-dimensional space. And you can see that they have differences in their state. So there's this distance that we can draw from in this morphospace. So A is where we have the morphological aspects of the OSS. The axes A1, A2, and A3 are shape coordinates that quantify different morphological aspects of the organoid. So these different axes here, A1, A2, and A3, are all shape components. So it's like if you take a microscopy image of a shape, you can break that down into different components. If you take like different measurements of shape, you can use a dimensionality reduction to get to these three axes that describe the variation. So there's a distance in this space. Then in B, you have a schematic representing the organizational aspect of the OSS. The axes B1, B2, and B3 are motif coordinates that quantify the cell type patterning in organoids. So this is where you have these motifs. You have, like in this case, you have sort of a annular shape of cells around a center mass of a different type of cell. In this case, you have a jumble of two different cell types. And in this case, you have partial ordering of cell types. And so they're all at different spaces in this, locations in the space along these three axes. So it's a variation on A, but it's describing things a little bit differently. I take it that these motif coordinates are ways to it's a different way of characterizing the phenotype. Single-cell mRNA sequencing in 3 of organoids enables the mapping of transcriptomic landscapes to define organoid state. So in 3, we have the organoid. We take the single-cell components of that organoid. We then map out the gene expression by cell for different genes, and then we get this transcriptomic landscape, which is this. They're using UMAP, which is a dimensionality reduction technique here, to show these pretty pictures of different, the different types of cells, different states, or the different types of cells. So you have 2 different progenitor types here, a ductal cell type and an endocrine cell type. And so they kind of divide out into different cell types using this kind of technique. And D, you see this for the organoid morphospace. So you can do the same thing with organoids and different features. You can extract features from this geometry. So each organoid has a specific geometry. You have the feature space and these different organoid IDs. And then you have the organoid morphospace with different states here. So there's, in this morphospace, which is distinct from the transcriptomic landscape, they're also using a UMAP visualization. You have these different features of an organoid, such as high curvature, multi-lumen, low curvature, and so forth. So you're getting these different components out of this complex geometric shape. And then in E, you have this mathematical modeling of this organoid morphogenesis process. It enables a mapping of the theoretical organoid state space. So here we have an organoid with different types of cells, in different cell states. You have these signaling centers. You have different physical descriptions such as contractility, pressure. You have ion pumps and tight junctions. So you have different functional aspects of the cells. You have different biophysical parameters and different biochemical parameters. And so you actually determine what the cell states are, what these parameters are, and then you model this mathematically into a space where you have 2 parameters and you have this variation, continuous variation in this space. So parameter 1 is that increases, there's a certain aspect of this organoid that changes. Parameter 2 increases, there's another aspect that changes, and then as they change together, you get changes in the shape. And so this is what they're doing here.
I guess I'm looking at it and saying, where's the adherence junctions on the outside of it?
Yeah, so that's a good question because they have to pick and choose what they're modeling, and so maybe they're not picking all the right things.
Right.
Yeah, but I guess it depends on what kind of substrate you build your organoid on. So organoids are usually they use a 3D scaffold or they use some sort of bioreactor where you have maybe no scaffold, but they do this in different ways. So yeah, but it would be interesting to see because most certainly the substrate plays a huge role in like what the organoids do.
Yeah, I'm sure it does.
Yeah. So Jesse says that so Dick says maybe we need a paper on the differentiation tree as an instantiation of the Waddington landscape. Yeah, I think so. That might be interesting because I know people have worked on like quantifying the Waddington landscape, but not like seeing how it resembles a tree so much. People talk about it being a tree because it's branching, but that it's like there's still a lot I think to kind of put into context there. The Waddington landscape really started as a sort of a not a literary device, but like as a very loose analogy. So like you put it on a landscape, it makes sense if you see a ball rolling down a landscape into canals, and it makes intuitive sense. But then people have tried to graft on like mathematical modeling and other things after the fact, and Waddington even suggested that there are these gene-gene interactions happening throughout the landscape, but that wasn't necessarily specified within the original conception. So that would be very interesting to see, to try to do that. Jesse says, sounds interesting. I continue to want to learn more about that kind of landscape development. Yeah, that would be good to follow up on. And
Yeah, I think cells are more fluid if they don't have adherence junction. I was reading something, and it said that in embryos before they hatch, the adherence junctions disappear, so that they can go out through a small space. At least that's what I was reading into it.
Yeah.
So that was an interesting thing.
Yeah. Well, yeah, I didn't go through the whole paper because it's pretty long, but I think you get the idea. So yeah, that's great. So I think that's all for today, unless anyone wanted to add anything else.
Well, I should get on with creating graphs. Yeah. That's my task for today, and tomorrow make a bunch of graphs.
All right. Well, thanks for attending. Have a good week.
Okay.
All right. Now I'd like to talk about two papers that are pretty exciting. One is on the liveliness of artificial life. So this is kind of a history and discussion about liveliness as a qualitative property of artificial life simulations. And then we're going to talk about a paper on C. elegans experimental evolution, which looks really interesting as well. So the first paper is on the liveliness of artificial life. This is Young, Colt, and Maurice Ling. And this is, I think they're from Singapore. And the abstract of this talks about this philosophical debate on whether artificial life models or digital organisms, whether they're truly alive or whether they just simply appear to be alive. So the main difficulty appears to be finding an encompassing and definitive definition of life. There was a book by Schrödinger in the 40s on this, and it was largely the basis for modern biophysics. There have been other definitions of life. In fact, the artificial life community has wrestled with this question since its beginning. The idea is, whether you can simulate life in a box in silicon, or whether it's something else that it can't be captured in modern digital computation. So this is where we are. This is a very hard philosophical question in terms of what is life, but then we have this idea of what are the identifiers of being alive. And like I said before, it's a qualitative question, but it's actually also something we can maybe make some progress on by examining similarities and differences in recent definitions of life. We define life as any system with a boundary to confine the system within a definite volume and protect the system from external effects, consisting of a program that is capable of improvisation, able to react and adapt to the environment, able to regenerate parts of itself or its entirety, with an energy system that compromises of non-interference sets, of secluded reactions for self-sustenance. It's considered a live or a living system. Any incomplete system containing a program and can be reassembled into a living system, thereby converting the reassembled system for the purpose of the incomplete system, are also considered alive. So that's a pretty broad definition, a pretty complete definition in some ways. Why don't we try to diagram this out and see what it looks like? So in diagramming this out, the first thing we notice is that they mention a boundary. So it might be something like this. We talked about boundary conditions with respect to organoids, but for now we'll just say it's a boundary, a physiochemical boundary that kind of gives us the working definition of a boundary. We go back to this, so any system with a boundary to confine the system within a definite volume. So we definitely have this boundary conforms to that definition. So this is a volume. And it could be a planet, it could be like an ecosystem or whatever. And protect the system from external effects. So our boundary is going to have maybe something like a gamma layer, which protects us from gamma rays, interstellar rays, an ocean where we're protected from predators, so we're on an island, those sorts of things. Consisting of a program that is capable of improvisation. So our boundary allows us to avoid some of these external effects. It's not a closed system, mind you, it's just that it's protective. So thermodynamically, it's an open system. And then consisting of a program that is capable of improvisation. So within this boundary, we have these agents that can do things that are improvisational. I guess what it means is they can behave in different ways. Sometimes they can make up things as they go along. They're not, restricted to a specific routine. They can change things. There's plasticity here. Now, we can also talk about a genome, which we'll talk about in a little bit. React and adapt to the environment, able to regenerate parts of itself or its entirety, with energy systems that compromise non-interference sets of secluded reactions for self-sustenance. So that last part is really long. But what they're getting at is maybe like two things here. There's a metabolism. All right, and then there is a generative capacity. And the regenerative capacity, you can see this sometimes in single cells or in maybe simple multicellular organisms, where if you ablate some aspect of this, it can regenerate its form, filling in the gaps. And there are different ways that this can happen. We won't get into that. This is very hypothetical at this point. But there's this regenerative capacity where if something is damaged or injured, it can re-establish itself. Sometimes this is through pattern formation. There are a lot of competing hypotheses as to how this happens. The more important part is the metabolism, because the metabolism in turn will drive this regenerative capacity. And so one of the things about metabolism, of course, is that you need independent cycles and independent reactions that come together. And so in early life, for example, you might have some independent reactions. Biochemical reactions form a cycle. Those cycles then are coupled in this organism. So the organism is incorporating these cycles into some larger function that does something for the organism. So this is where we have these secluded reactions for self-sustenance. So these are non-interference sets here, but they do interact in a single organism. Any incomplete system containing a program and can be reassembled into a living system, thereby converting the reassembled system or purpose of the incomplete system, are also considered alive. And this, of course, is where you have a complete system containing a program. So in this case, we would have an organism or a cell with a genome. And that genome would contain instructions, maybe to take like a cell that needs to regenerate. So we have our cell that's broken, it needs to regenerate itself. It has a genome with instructions on how to do that. That genome is put into the world, it's expressed, and it rebuilds the cell. So something like that is considered alive. You could also have a genome that was, needs to reproduce itself. So you have, you can have cell division. So you can have clonal cells, you can have offspring, eggs, things like that, that all have a copy of the genome in it, and they reproduce. So replication is part of this too. And that puts, that gives you this last criterion of having a reassembled system from an incomplete system. And, classifying a replicator like that is kind of odd, but they don't mention replication in here, so I don't really know why they kind of skirted around that requirement, because it seems like replication, where having a replicator is a very important part of being alive. In any case, so that's their definition. And of course, then you say, okay, if I simulate that in a computer, if I take those things and I build a system, model those things, does that give me something that's alive? It may look alive, but is it alive? And this is kind of a hard question to ask, because, you're sort of asking maybe something that can't be answered. I mean, it's up to the eye of the beholder. I might think it's alive. You might not think it's alive. It behaves like it's alive, but it's in a computer. You might think that criterion alone is something that does not make it alive. That doesn't necessarily matter, though, definitionally. So, using this definition, we argue that digital organisms may not be the boundary case of life. So, digital organisms may not be where sort of life ends, even though some digital organisms are not considered alive, thereby taking the view that some form of digital organism can be considered alive. In addition, we present an experimental framework based on continuity of the overall system and potential discontinuity of elements within the system for testing future definitions of life. So, there's been this question of like, what is alive? And so, you can ask people, are viruses alive? And so in an introductory course to microbiology, if you ask this question to those undergraduates, 79% of the students change their opinion before and after you consider the question and taking the course, compared to genetic engineering, which has a 56% opinion swing. I think the point is that a lot of people, once they understand the problem and they see something like a virus and how it behaves, and they look at other things and they make that comparison in their head, that they change their mind. Using boundary cases classification criterion, also known as the decision boundary or decision surface, has been well established. The reason that all of our virus is alive makes an interesting debate is simple. We will have established certain criterion, certain definite criterion separating what is alive from what is not by answering this question. But of course, that's only an opinion. That's not really our core criterion. And, maybe we can do something like a test of liveliness, which, it puts you down the road of something like a Turing test, but it's not necessarily that. And I don't want to get into that sort of thing, because Turing tests are hard to really kind of prove anyways. So, using tools of molecular biology to study the genetics of viruses, the debate continues. So it's hard to say that we're going to solve that problem in the near future. But in the artificial life world, people have asked these questions as well. And so, in 1986, Christopher Langton coined the term artificial life. This is kind of the first steps towards this sort of robust A-life field we see today. And this is where, the general view taken by A-lifers, artificial life researchers, is that carbon-based biological life on Earth is merely one manifestation or implementation of life. So if your attitude is that all life has to be carbon-based, you might have silicon-based life on another planet or a moon somewhere. You might have other types of life. We don't know. And the consideration in A-life is what is a very inclusive definition of life that goes beyond carbon-based biological life. So, and even in the early days of Earth, at the origins of life, we may have had very different types of things that were going on that constituted life. So we maybe, had macromolecules that were silicon-based instead of carbon-based. There are all these cycles, these independent cycles that I talked about in the organism that rely on these sorts of substrates. So in a silicon-based life form, you have these types of silicohydrates. In the carbon-based life form, you have carbohydrates. And they're using maybe this similar pathways, but they're using them in different ways. But then the other thing is, are computer simulations that look alive, are they alive? And so this is the problem. And some people would not consider that just simply because there's this, maybe this bias towards saying something in a computer is alive, but nevertheless, it looks alive to a lot of biologists sometimes, but it's not indeed alive. So, without getting too deeply into this philosophical argument, one of the things, we have a lot of definitions of life. Macklem and Seeley define life as the self-contained, self-regulating, self-organizing, self-reproducing, interconnected, open thermodynamic networks of component parts existing in a complex regime which combines stability and adaptability in the phase transition between order and chaos, which is basically saying that these organisms bring themselves into existence. So these biochemical cycles come together, and there's a boundary drawn around the environment it's in. There's a boundary drawn around the organism. If you think about eukaryotic cells, they emerged from the incorporation of numerous organelles that were actually independent organisms before the advent of eukaryotes. These things self-organized into a single container. They were able to do thermodynamic work, so they were contained there, but they were also open thermodynamic networks, which means that they were able to have free energy coming from the outside, do work within themselves, and produce and sustain life, and then sustain their own life. And of course, then they were islands of entropy production, so they would dump their entropy outside the organism, but inside they were minimizing entropy. The last thing I wanted to get into in this paper is that it goes through a very lengthy set of definitions and when they were proposed. And the first one they have is from the year 1855. And so this is where, you go through and you see some of these. Bernard, who of course did a lot of work on homeostasis and the internal milieu in physiology, is interested in self-regulating internal states. His definition, unfortunately, was, if I had to define life in a single phrase, I should say life is creation, which is of course useless because what is life? It's a creation. What is creation? And then that's where you get into this, circular sort of logic of trying to prove creation. And so we know creation is more in a more detailed sense today, but I think in his time, this was something that is not really that well defined. So yeah, and then we have all sorts of people here: Engels, Spencer, Putter, von Bertalanffy, and so there's some people from cybernetics in here. Von Bertalanffy, Bernard, I guess, would be considered cybernetics. Schrödinger in What is Life in 1944, life is replication plus metabolism. Replication is explained by the quantum mechanical stability of molecular structures, while metabolism is explained by the ability of a living cell to extract negative entropy from its surroundings in accordance with the laws of thermodynamics. Of course, thermodynamics is a big focus because he was a physicist and he wanted to think about this in biophysical terms. Von Neumann proposed life is not one thing but two: metabolism and replication. They are logically separable. So metabolism is separable from replication, at least logically. So that's an interesting definition, and one that maybe we can revisit that because I think that's kind of an interesting concept. Oparin, who of course did a lot of work on the origins of life, any system capable of replication and mutation is alive. And of course, in his context, that was true. That was basically the dividing line between life and non-life. Von Bertalanffy also gives another definition in 1968, takes another stab at it. Life is a hierarchical organization of open systems. So this is where we have these open systems that have a boundary, but they're also thermodynamically open, and we have this hierarchical organization, meaning that these partitions, they're sub-partitions under sub-partitions. So you have these structures like, you might have an ecosystem, you might have a niche, you might have an organism. That organism has organs. Those organs have some organization within them that favors, order. So this is kind of an interesting definition. If you go to things like hierarchy theory and other types of ecologically based theoretical biology, you'll see this. You'll also see this in a lot of the literature on self-organization as well. So Maturana and Varela give a definition. Life is a metabolic network within a boundary. So this goes back to our diagram of the organism with its separable metabolic cycles and how they're linked together in a common organism. So life is a metabolic network with a boundary. All that is living must be based on autopoiesis. And if a system is discovered to be autopoietic, that system is defined as living, must correspond to the definition of minimal life. So there's a definition of minimal life, something then, if it satisfies that, it's autopoietic, and then it's got another set of definitions that go along with that. So it's kind of an interesting way to do that. Ilya Prigogine in 1980, living units are viewed as objects built up of organic compounds as dissipative structures, which are these structures that form around free energy, like a river. The flow of a river will bring different types of plants and animals around it, and then when the river dries up, those go away. So it serves as an organizing mechanism for energy, or at least dynamic low entropy systems, significantly displaced from thermodynamic equilibrium. So Prigogine saw life as being out of equilibrium, and that kind of goes back to this idea of these systems that have these boundaries but also are open, and with organisms being sort of these entropy minimizing systems. So, again, we go back to thermodynamics. And of course, the idea of open systems is related to thermodynamic equilibrium, because thermodynamic equilibrium is something we associate with a closed system. If it's far from equilibrium, that means you get a lot of energy pulses and a lot of fluxes that allow for these systems to persist. And so there are a lot of different definitions over time. As we get into the modern era, they get longer, it seems like. Some of them not so much, but the NASA working definition of life, which was defined in 1994, is life is a self-sustained chemical system capable of undergoing Darwinian evolution. So we have this idea of replication and selection, natural selection, and then we also have this idea of just a self-sustained chemical system. So a hypercycle, if people are familiar with that, that was something that Eigen and Schuster came up with in the 70s, I believe. This is where they built these hypercycles, these chemical cycles that were self-reproducing, self-modifying, and they didn't really need to be in an organism. They could just be chemical hypercycles. And so that might be a definition of life as well. Ruth Bernstein and Dillon, and these are physiologists, they're talking about, in 1997, their definition is living organisms or systems characterized by being highly integrated through the process of organization driven by molecular and higher levels of complementarity. So this is good paper. It has a lot of definitions of life, so we can go back and look at that if you're interested. The second paper is on selection and direction of phenotypic evolution. And this is by a group in Paris, Millard, Alfonso, and Teotonio. And they're interested in doing these experimental evolution with C. elegans, and they're interested in looking at the direction of selection and phenotypic evolution. So we'll see what that looks like in a bit. It's basically a map where it shows a direction of evolution relative to the state of the current organism, or the baseline organism. So the abstract is predicting adaptive phenotypic evolution. Depends on invariable selection gradients and on the stability of genetic covariances between the component traits of the multivariate phenotype. So, if we're trying to predict evolution, we need to have some sort of idea of the selection gradient. It needs to be constant, and then we need to have covariance between different genes. So if we know that different genes make up an organ or make up a phenotypic trait, those covariances need to be high, so they need to evolve together. And then we also have to have not a lot of variation in selection. Selection can vary over time, so we want to have a constant source of selection. And if we have that, then we can predict the way that a phenotype will evolve. If we don't have that, then we can't necessarily do that as well. We describe the evolution of 6 traits of locomotion behavior and body size and the nematode C. elegans for 50 generations of adaptation to a novel environment. So they did a 50-generation evolutionary experiment. We show that the direction of adaptive multivariate phenotypic evolution can be predicted from the ancestral selection differentials, particularly when the traits were measured in the new environment. And so they were taking their C. elegans, they had a baseline organism, and they put it into this environment where this was served as the treatment. They basically allowed the different populations, maybe the population under selection pressure and the population not under selection pressure, to reproduce. They would select the most fit offspring. They would then go to the next generation and so forth. And the idea is that being exposed to this environment, I think it was a salinity gradient, would select for the organisms that were most fit for that. The selection environment was constant because you would always have a constant concentration of the treatment, and then you would just keep doing this for 50 generations, and you would end up with a different set of traits, or the traits that have evolved. We show that the direction of adaptive phenotypic evolution can be predicted. Interestingly, the evolution of individual traits does not always occur in the direction of selection. So the evolution of traits don't occur in the direction of selection, so you could select for organisms that survive, but the traits underlying that don't necessarily line up with selection. So you're selecting for something and you get more variation than you would expect in a constant environment. You might expect that individual traits evolve with the direction of selection, but that's not what they found. Nor are trait responses to selection always homogeneous among replicate populations. So, one of the reasons is because the responses to selection are heterogeneous. You have different pathways that might be used. You have different genetic backgrounds, even in C. elegans, where they're not clonal, but they self-reproduce. And so you have this aspect of where there's a lot of variation within the response to, say, a simple stimulus. These observations are explained because the phenotypic dimension with most of the ancestral standing variation only partially aligns with the phenotypic dimension under directional selection. So the standing genetic variation is only partially aligned with the phenotypic dimension, meaning that you have genes that are, not connected to the trait. And complex traits often have many genes, a lot of them with low levels of explanatory power, so they're not necessarily, they don't explain all the variation in the phenotype. So you get things like that change, and that's what introduces a lot of this variation. These findings validate selection theory and suggest that the direction of multivariate adaptive phenotypic evolution is predictable for tens of generations. So they went to 50 generations in this, and they showed some of their results. Now, this does not mean that you can predict for hundreds and hundreds of generations. So I'm going to go to the figures. So they talk about this, they're doing experimental evolution in high salt environments. So they're exposing the C. elegans to salt concentrations, which are much higher than what they normally experience. And so it's thought that if you have the ability to a tolerance to high levels of salt, that you are selected for, and this happens over 50 generations, so it should filter out a lot of the organisms that don't have a tolerance to salt. But the thing that they mentioned in the abstract was that there may be many ways you can get to salt tolerance. It's not always the same pathway. So, this is something that the organisms will exhibit some plasticity here. They don't always use the same mechanisms. So this just shows like some of the genetic variants here, the genetic covariances. So low salt and high salt. So for low salt, you have you have some positive covariances, some negative covariances, very relatively low, negative 0.2 to positive 0.2. And so this just shows some of these. This shows alignment between phenotypic plasticity and standing genetic variation. So there's some variation between the two. This is null in the observed. This is null, and then some of the examples. This is from the ancestral G matrix. So this is another analysis graph. This is where you have selection gradients here, and you have these different FS and BS traits. So they define that in the paper. The gray lines are the from the ancestral high salt G matrix with low genetic variance, and then the red lines are without low genetic variance. So this is a comparison of those two. So for these different traits, you see that there's these differences. And there's some difference in terms of more or less aligned, but you do have this variation here. So you can see that there's, first of all, the error bars are somewhat large, and then secondly, they behave differently with respect to the selection gradients. So that's all for today. Thank you for paying attention, and I hope you learned something.